Welcome to the Deep Sleep Science channel. Tonight, we're diving into one of the quietest and most mesmerizing phenomena in all of nature light that is alive. Imagine an ocean so deep that sunlight never reaches it, yet it is not truly dark. Tiny flashes ripple through the water like distant stars. Creatures drift through blackness, glowing softly, signaling, hiding, hunting, all without a single ray of the sun. And it's not just the ocean. In forests, you might find wood that seems to glow gently in the dark. In caves, ceilings shimmer with living lights. In summer nights, entire fields pulse with fireflies speaking in silent patterns. What makes this even more fascinating is that these lights are not reflections, not fire, not electricity, but chemical reactions happening inside living bodies. Nature has learned over millions of years how to create light itself again and again in completely different ways. Some creatures use it to disappear. Some use it to attract. Some use it to deceive. And some glow without ever moving at all. Tonight, we'll slowly explore how life learned to shine in the darkness, from microscopic plankton that turn waves into glowing oceans, to deep-sea predators that carry their own private lanterns, to organisms so small, and so subtle that their light is only revealed when the world around them moves. So as you settle in, let your mind follow these quiet lights drifting through water, forest, and night, and gently discover the science behind one of the most peaceful and beautiful mysteries on Earth. Bioluminescence is not one single trick used by one small group of organisms. It is a vast natural phenomenon that has appeared again and again across life, in very different environments and in very different forms. A glowing wave, a flashing firefly, a luminous shark, and a faintly shining fungus are all part of the same broad idea: living things creating their own light through biology. The deep ocean contains such an enormous volume of living space that bioluminescent light may be the most common form of visual signaling on Earth. In the dark waters far below the reach of the sun, many animals rely on light made by their own bodies or by helpful microbes. In a place where sunlight fades away, these small flashes and glows become one of the main ways life communicates, hides, hunts, and survives. In open ocean waters below about 500 meters, bioluminescence is estimated to occur in about 90% of animals. That means in many deep marine environments, glowing is not unusual at all. It is part of the basic visual background of life there, almost like an everyday language of light. Researchers at Mbari have reported that more than 3/4 of animals drifting in the water column can create their own light. This tells us that bioluminescence is not a strange side feature of the deep sea. It is woven into the ordinary lives of many midwater creatures, from delicate drifting forms to active predators. Marine bioluminescence appears in organisms ranging from bacteria and algae to sharks and fishes. That wide range matters because it shows how broadly useful light can be in the ocean. Very small life forms use it, and much larger animals do too, each in a way that suits its own body and way of life. Fish are the only vertebrates known to include naturally bioluminescent species. That makes them stand out among animals with backbones. While mammals, birds, reptiles, and amphibians are not naturally bioluminescent, some fishes have evolved the ability to glow, especially in the dark ocean where light can mean the difference between being seen and staying hidden. In fish alone, about 1/500 known species can luminesce. That is a remarkable number, and it shows that glowing fish are not rare exceptions. Instead, light production has become an important feature in many branches of fish evolution, especially in the deep sea where visual cues remain useful even in darkness. The ocean's darkness did not shut down visual evolution. In many ways, it encouraged new forms of it built around self-made light. Instead of depending only on sunlight, deep-sea creatures evolved ways to produce their own signals, turning darkness into a setting where even a tiny glow could carry great meaning. Bioluminescence can be defensive, deceptive, cooperative, reproductive, or simply useful for hiding, depending on the organism. A single kind of light may mean danger in one species, attraction in another, and camouflage in a third. That variety is part of what makes this topic so rich. The glow is only the beginning, and the purpose changes with the creature. One major strategy is counter-illumination, where an animal glows on its underside to match faint light from above and erase its silhouette. In the open ocean, an animal seen from below can look like a dark shape against dim surface light. By shining softly downward, it can blur that outline and become harder to detect. Counter-illumination is especially useful in midwater habitats where hiding places are scarce. There are no rocks, no caves, and no plants to slip behind in much of the open sea. So instead of hiding behind something, some animals hide by controlling how they appear against the light itself. Another important strategy is the so-called burglar alarm effect. In this case, a small animal under attack flashes brightly enough to attract an even larger predator to the attacker. The light is not aimed at winning the fight directly. Instead, it shifts the danger, turning one predator into a target. Some species use light to attract prey closer. In darkness, even a tiny glow can look like food, movement, or a possible opportunity. A predator can take advantage of their curiosity, drawing prey near enough for a quick strike. Other species use bioluminescence to detect or illuminate prey in darkness. Rather than just acting as a lure, the light can help reveal what is nearby. In a place where natural sunlight cannot help, organisms sometimes make their own brief flashlight. Many organisms also use light as a communication signal to members of their own species. A glow can help an animal locate a mate, identify its own kind, or coordinate with others nearby. In that way, bioluminescence becomes more than a physical reaction. It becomes a message. Some animals can use the same display for several purposes at once. A light might work as camouflage from one angle, as a lure in another moment, and as a social cue under different conditions. This layered usefulness helps explain why bioluminescence has evolved so many times. The deep sea favors light-based traits because there is very little sunlight to compete with them. In bright surface environments, a faint signal can be washed out. But in darkness, even a small flash becomes highly noticeable, which makes light a very powerful tool. In some deep habitats, a single emitted flash may be the most visible event in the area for several meters around. That gives light extraordinary importance. A brief glow can suddenly reveal a living presence in otherwise nearly complete darkness. Because many deep-sea animals make light, some fishes have evolved ultra-black skin to absorb bioluminescent light and avoid detection. Instead of shining, these animals survive by doing the opposite. They reduce reflections so effectively that even when light falls on them, very little bounces back to reveal their shape. That ultra-black skin can reflect less than 0.5% of incoming light. In practical terms, that means these animals are exceptionally difficult to see. In the deep ocean, survival may depend not only on making light skillfully, but also on disappearing from the light made by others. Bioluminescent waves caused by dense blooms of dinoflagellates are one of the best-known surface displays of living light. At night, disturbed water can flare into electric blue lines and sparkles. It is one of the rare times when a phenomenon driven by countless microscopic organisms becomes visible on a grand, sweeping scale. Boat wakes in bioluminescent water can appear outlined in light, because each disturbance triggers countless tiny flashes. Instead of a single glowing organism making a large display, the shape of the moving boat is drawn by a crowd of microscopic lives reacting together. The effect can make the sea look as though it is sketching motion in light. The same principle can make fish or dolphins appear edged in blue as they move through glowing plankton. Their bodies do not create that light directly. Rather, their movement sets off a response in the organisms around them. So the animal seems to carry a luminous outline through the water. Dense dinoflagellate blooms show that bioluminescence can become a large-scale landscape phenomenon, not just an individual trait. The light is still produced cell by cell, but when enough organisms gather, entire shorelines can appear to pulse and shimmer. Tiny life can create a scene that feels planetary in scale. Bioluminescence is light produced by a living organism through a chemical reaction, not by reflection and not by heat. This is one reason it feels so unusual. It is not borrowed light, and it is not the glow of something burning. It is built directly by biology. The process is a form of chemiluminescence that happens inside living tissue. In simple terms, specific molecules react with one another and release energy as visible light. The glow is part of a chemical event, but one that has been shaped and refined by evolution. In many bioluminescent systems, a molecule called luciferin reacts with oxygen to help generate light. Luciferin is one of the central chemical players in many glowing organisms. When it takes part in the right reaction, energy is released in the form of visible light rather than mostly heat. That reaction is often accelerated by an enzyme called luciferase. The enzyme helps the chemistry happen efficiently and at the right speed. In living systems, enzymes often guide reactions with precision, and here that precision allows a creature to turn chemistry into light. Some organisms use photoproteins instead of a standard luciferin-luciferase pairing. This means bioluminescence is not built the same way in every lineage. Nature has found more than one chemical path to the same broad result: a glow in the dark. A photoprotein can emit light when it combines with an oxidizing agent, and in some cases it does not need a separate catalyst enzyme. This is another reminder that bioluminescence is a collection of systems, not one universal recipe. Different organisms may glow for similar reasons while using different underlying chemistry. Many bioluminescent organisms produce what is called cold light, meaning very little energy is wasted as heat. This is one of the most elegant parts of the phenomenon. The reaction is efficient enough that the organism gains a visible signal without creating much unnecessary warmth. Bioluminescent light in the ocean is usually blue or blue-green. That color pattern is not random. It reflects the physical reality of seawater, which allows those wavelengths to travel farther than many others. Blue-green light dominates marine bioluminescence because it moves through seawater more effectively than red light. In the ocean, distance matters. A signal that travels farther can be more useful for attraction, warning, or communication. So evolution repeatedly favors the colors that work best in water. Although blue is most common, some organisms produce nearly violet, green-yellow, or even red bioluminescent light. This color diversity shows that evolution is flexible. While blue is usually the practical choice in marine settings, different ecological pressures can push species toward more unusual colors. Bioluminescence is relatively rare on land compared with the sea. That contrast is striking. The ocean, especially the deep ocean, is full of glowing life, while on land the phenomenon appears in more limited and specialized groups such as fireflies, glowworms, and some fungi. In the ocean, bioluminescence is common enough to be considered one of the defining features of deep pelagic life. When scientists describe the biology of the deep open sea, light production is not a side note. It is one of the central adaptations that shapes how creatures live. True plants are not known to produce natural bioluminescence. That is useful to remember because many glowing scenes in nature may look plant-like at first glance. But natural bioluminescence belongs instead to certain microbes, fungi, and animals, not to ordinary green plants. Natural bioluminescence is also not known in amphibians, reptiles, birds, or mammals. So although many vertebrates are familiar to us on land, the natural production of living light remains absent from those groups. This makes glowing fish even more unusual among animals with backbones. Surface bioluminescence reminds us that not all glowing ocean life is deep-sea life. While the deep ocean is especially rich in luminous creatures, some of the most visible displays happen much closer to shore, where human eyes can watch the water respond to waves and movement. Dinoflagellates are tiny single-celled plankton, yet they can produce behaviorally meaningful light. Their small size does not limit the impact of their glow. In fact, their collective flashes create some of the most famous bioluminescent scenes in the world. When dinoflagellates bloom densely, ocean water can look reddish-brown by day and then sparkle at night when disturbed. This day-and-night contrast is one of the most fascinating parts of their behavior. The same water can appear calm and murky in sunlight, then suddenly alive with blue flashes after dark. Waves, boat wakes, swimming animals, and even a hand moving through the water can trigger dinoflagellate flashes. The glow is often a response to physical disturbance. It is as though motion itself writes light into the surface of the sea. Their flashes are usually brief, which makes bioluminescent water glitter rather than shine in one continuous beam. That rapid on and off response gives the sea a sparkling quality, more like scattered points of light than a steady lamp. Some plankton are not equally ready to glow at all times of day. In the dinoflagellate Pyrocystis fusiformis, the chemicals involved in light production are made at dusk, guided by an internal biological clock. So even microscopic light can be tied to timekeeping inside a living cell. Not every red tide is bioluminescent, and not every bioluminescent bloom is toxic. These two ideas are often linked in public imagination, but they are not the same thing. A glowing coastline may be beautiful without necessarily indicating toxicity, and a red tide may exist without producing a night glow. Some organisms do not make light entirely on their own, but rely on symbiotic bacteria. In these partnerships, the host provides shelter and nutrients, while the microbes provide the glow. This makes bioluminescence not just a matter of chemistry, but also of cooperation between living partners. The Hawaiian bobtail squid is a famous example of this kind of partnership. Soon after birth, its light organ can be colonized by the bacterium Vibrio fischeri. The squid then uses that bacterial light for camouflage, glowing downward so that its shadow is reduced when seen from below. Female deep-sea anglerfish also show how powerful bacterial partnerships can be. Their glowing lure, called an esker, sits at the tip of a modified dorsal spine and can contain luminous bacteria. In the darkness of deep water, that tiny glowing structure acts like a baited signal suspended in front of the predator's mouth. Jellyfish are among the most famous marine animals associated with bioluminescence, and one species, Equoria victoria, changed science. Work on its light-producing system led to the discovery of GFP, a protein that later became essential in biology labs for tracking cells and proteins. In Equoria victoria, the protein equorin emits blue light, but the jellyfish appears green because another protein, GFP, absorbs that blue light and re-emits green fluorescence. This means one of the best-known natural glows depends on both bioluminescence and fluorescence working together in a single optical system. The original glow of that jellyfish became far more than a marine curiosity. GFP was later used by scientists to observe gene expression, track proteins, and study living cells in real time. A soft green light from a jellyfish ended up helping transform modern biology. Bioluminescence has evolved many times independently rather than coming from one single glowing ancestor. This repeated evolution tells us that making light has been useful in many different ecological settings. Whenever darkness, signaling, camouflage, or attraction created the right conditions, life found new ways to glow. The widespread appearance of bioluminescence across bacteria, protists, fungi, and animals is strong evidence of repeated evolutionary invention. Different groups arrived at light through different histories and sometimes different chemistry. What unites them is not shared origin alone, but the repeated value of light as a survival tool. The study of bioluminescence keeps expanding because every newly studied glowing organism may reveal a new chemistry, a new behavior, or a new ecological relationship. The glow that appears so simple at first often leads to deeper questions. And in that way, bioluminescence remains one of the clearest examples of how nature can turn invisible chemistry into visible life. One reason surface bioluminescence feels so magical is that it often happens close enough for people to witness directly from shore. Dinoflagellate light shows are among the most familiar examples of marine bioluminescence, for exactly that reason. Unlike many deep-sea displays, these glowing events happen near the surface, where waves, wakes, and movement can bring the light into view. Southern California has become especially well known for bioluminescent waves linked with red tide events. In those moments, the surf can glow in electric-looking blue as water is disturbed. It is a reminder that some of the most memorable encounters with living light happen not in remote deep water, but along coastlines where microscopic organisms gather in enormous numbers. Some bioluminescent red tides are thought to involve dinoflagellates using light as a way to avoid predators. The flash may help startle a grazer or call attention to it in a dangerous way. So even a beautiful shoreline glow can be rooted in a survival strategy taking place at a scale far too small for the eye to see directly. The dinoflagellate Noctiluca is associated with glowing blooms in some regions of the world. It stands out because it is not just a passive drifting cell. It is an active participant in the food web, and its blooms can become large enough to change how coastal waters look by day and by night. Noctiluca is a predator rather than a classic photosynthetic plankton cell. That makes it different from the more familiar idea of plankton as tiny drifting organisms powered mainly by sunlight. Its role shows that bioluminescent plankton are not all doing the same kind of biology, even if they contribute to similar glowing events. Scientists describe Noctiluca as capable of rapid growth that can lead to blooms. Once conditions become favorable, populations can increase quickly, turning what began as countless invisible individuals into a large-scale visible phenomenon. This rapid expansion helps explain why glowing coastlines can sometimes appear dramatically and seem to transform whole stretches of water. Marine bioluminescence visible from beaches often comes from organisms too small to see one by one. To the human eye, the light seems to come from the water itself. But in reality, it is the combined reaction of huge numbers of microscopic lives, each producing its own tiny flash. This makes bioluminescent surf one of the few natural light displays created by enormous crowds of microscopic organisms acting independently. There is no central coordination and no single source. The broad glow emerges from countless separate responses, all triggered by movement in the water. Dinoflagellates are an example of bioluminescence in a protist rather than an animal. That matters because it widens the story of living light beyond creatures with nerves, muscles, and complex bodies. Even single-celled organisms can evolve systems that create meaningful, functional light. Their glow is often strongest in calm coastal conditions that allow blooms to collect near the surface. If the water is stable enough for these cells to gather in large numbers, then even ordinary wave motion can reveal them dramatically. So the brightness people observe often depends not only on the organism, but also on the physical structure of the water. Red tides show that a bioluminescent event can also be part of a harmful algal bloom system, even though the light itself is not the harm. This is an important distinction. The glow is a visible effect, but the environmental and biological consequences of blooms depend on the species involved and the chemistry of the event, not simply on the fact that the water shines. In many luminous partnerships, the host animal provides shelter and nutrients, while bacteria provide the light. This arrangement is elegant because each partner contributes something the other needs. The host creates a stable home, and the microbes generate a steady glow that the host can use for survival. Anglerfish are famous examples of animals that house glowing bacteria in a specialized organ. The relationship looks simple from the outside, but it depends on careful control. The host has to maintain the right structure and environment so the bacteria remain useful rather than becoming just another microbial population. The glowing lure of an anglerfish is called an esker. This small structure is one of the most recognizable pieces of anatomy in deep-sea biology because it turns the front of the fish into something like a living fishing tool. In darkness, a tiny point of light can be enough to invite a fatal approach. The esker sits at the tip of a modified dorsal spine called the elysium, which works almost like a flexible rod. This arrangement allows the fish to position the light in front of its mouth rather than simply glowing from the body. That placement matters because it turns the light into a controlled lure instead of a general body signal. In many anglerfish, the bacteria inside the esker belong to a single species rather than a random mixture. That suggests the partnership is not accidental. It is a refined biological relationship in which both host and microbe are matched in ways that support reliable light production. The light from the esker helps draw prey toward the anglerfish in otherwise black water. In the deep sea, curiosity can be deadly. A prey animal investigating a faint glow may be moving straight toward a predator that is barely visible behind it. Anglerfish show that vertebrates can effectively outsource light production to microbes. Instead of evolving every part of the chemistry alone, the fish depends on living partners. This expands the meaning of bioluminescence, showing that in some animals the glow is the result of a small, internal ecosystem. The bobtail squid forms another famous partnership with luminous bacteria, and this begins very early in life. The light organ can be colonized within hours of birth, showing how quickly the animal starts building the relationship. The glow is not an optional extra added later. It becomes part of the squid's life, almost from the beginning. The bacterium best known from the bobtail squid relationship is Vibrio fischeri. This microbe has become one of the classic examples used to study how animals and bacteria cooperate. Its role in the squid is so well known that the partnership is now one of the standard biological models for symbiosis. The bobtail squid uses bacterial light mainly for camouflage rather than for luring prey. This is an especially beautiful solution to the problem of being seen from below. Instead of trying to hide by becoming darker, the squid hides by glowing in a carefully controlled way. By shining downward, the squid can match the faint light coming from above and reduce its shadow. In open water, a predator looking upward may notice a dark outline. But if the squid produces just the right amount of light, that silhouette can soften or disappear into the background. This is one of the clearest examples of counter-illumination in nature. The animal does not merely glow for show. It uses light with precision, adjusting appearance in relation to its surroundings, so that being luminous actually helps it become less visible. Some squid species, besides the bobtail squid, also host luminous bacteria in organs called photophores. This shows that bacterial bioluminescence is not limited to one famous species. Similar solutions have evolved in multiple lineages where controlled light offered a strong advantage. Flashlight fishes also rely on symbiotic bacteria in specialized light organs. Their glow is positioned in a way that makes it especially noticeable, and the system is built for repeated use. These fishes demonstrate again that anatomy and microbial partnership can work together to produce a highly functional living lamp. In flashlight fish, the light-producing bacteria are packed into tubules inside the organ. This means the host is not just carrying microbes loosely. It is organizing them in a structured biological space that supports efficient, directed light output. In the fish anomalops, those tubules are aligned at right angles to the organ's surface. That kind of alignment suggests that the organ is engineered by evolution for optical performance. The bacteria make the light, but the host body helps shape how that light leaves the animal. Flashlight fish can use their glowing organs during nighttime schooling behavior. In darkness, coordinated light may help neighbors keep track of one another, remain grouped, or move together more effectively. So the glow may support social organization as well as visibility. Bacterial symbiosis allows some animals to gain light without having to make every part themselves. From an evolutionary point of view, this is a powerful option. If a useful trait can be achieved through partnership, natural selection can shape the relationship instead of building the chemistry from scratch inside one lineage. These relationships also require the host to control bacterial populations carefully, so the light remains useful rather than wasteful. Too little light would fail to help, and uncontrolled microbes could create biological problems. The host must regulate the system in a way that keeps the partnership stable. A host must maintain the right anatomy as well, because ordinary tissue does not automatically function like a lamp. To make a luminous organ useful, the body often needs chambers, surfaces, and structures that position, direct, or control the glow. Reflectors and lenses can be part of these organs, making them more like biological lighting devices than simple glowing patches. This means some animals do not merely emit light, they manage it. They shape the beam, intensity, and direction in ways that resemble the design principles of human-made optics. Symbiotic systems show that bioluminescence is not only a chemistry story, but also an ecological one, involving cooperation between species. The glow depends on interaction, balance, and mutual benefit. In that sense, some luminous organs are not just traits of one organism, but shared systems. The same kind of bacterial partnership can support camouflage in one organism and predation in another. This is fascinating because it shows that the meaning of a glow depends on context. The bacteria may be producing light in similar ways, but the host uses that light for very different purposes. Mutualistic bioluminescence can become so central to survival that the light organ turns into a major developmental feature. The animal's body plan ends up shaped around the partnership. This tells us the relationship is not shallow or temporary, but deeply built into how the organism grows and functions. The bobtail squid system is widely used as a model for studying animal microbe symbiosis. Scientists are interested in how the squid recognizes the correct bacteria, how the organ develops, and how a beneficial relationship is established so early and so reliably. Because the bacteria gain shelter and nutrients, the relationship is helpful to both partners. The host gets camouflage, and the microbes get a secure place to live and grow. That balance is one of the central reasons the partnership can persist through evolutionary time. Bacterial bioluminescence proves that an animal's glow is not always genetically owned by the animal alone. Sometimes the visible signal comes from another species living inside it. That idea softens the boundary between individual and partnership, at least when it comes to traits like light. Some of the most iconic glowing animals are therefore really miniature ecosystems. What appears to be one luminous creature can actually be a coordinated collaboration between host tissues, symbiotic microbes, and specialized anatomy, all working together. Jellyfish-like animals are another major part of the bioluminescent world. Comb jellies, siphonophores, and many other gelatinous forms contribute heavily to the visual environment of the midwater ocean. In some deep habitats, their glow is so common that it helps define what darkness looks like there. Some deep-sea jellyfish use luminous displays as defense, creating sudden patterns that distract attackers. The glow may not injure the predator directly, but it can interrupt the moment, produce confusion, and create a chance for escape. Even a delicate-looking animal can use light in forceful ways. Comb jellies, also called ctenophores, are frequent contributors to deep-sea light displays. Their bodies are often transparent and fragile, yet their light can move in striking patterns. The combination of softness and signal makes them some of the most visually remarkable luminous animals. In ctenophores, waves of light can travel across the body. This moving display can make the animal look as though it is lit from within by traveling currents. The pattern is not just beautiful. It reflects how bioluminescence can be dynamic and spatially organized, rather than fixed in one point. Some of the most spectacular luminous forms in the deep ocean belong to siphonophores, which are colonial animals rather than single-bodied individuals. A siphonophore is made of many linked units working together, so its light can be distributed through a larger, more complex structure. Because siphonophores are colonial, their bioluminescent displays can be coordinated across many zooids in one drifting body. This gives them a scale and complexity that differ from single-organism flashes. The light is part of a collective body architecture. Glowing gelatinous animals are common enough in the deep sea that many submersible observations include them. Their abundance means they are not just occasional curiosities. They form a substantial portion of the organisms shaping how deep marine darkness is interrupted by light. Although the glow of jelly-like animals may appear delicate, it often serves serious survival functions, such as startling predators. The softness of the body does not mean softness of purpose. In evolutionary terms, even a fragile animal can carry highly effective defenses. Because gelatinous animals are so abundant in midwater habitats, their light makes up a large share of the deep ocean's visual environment. If one could see the deep sea the way its inhabitants do, much of the available light might come from drifting, glowing, gelatinous bodies, rather than from fixed sources. Scientists often study these fragile animals with remotely operated vehicles because NEPs can damage them badly. This matters for understanding bioluminescence, because an animal that is torn or compressed may not show its natural behavior properly. Gentle observation is often necessary to understand how the light really works. Some jellyfish glows become visible only when the animal is disturbed. In those cases, the light is not a constant feature, but a triggered response. The animal may appear nearly invisible until some event causes it to flare into view. The light in gelatinous animals often spreads through transparent tissue, making the whole body seem illuminated from within. This creates a very different visual effect from a single point-like lure or organ. Instead of a lamp hanging from a body, the body itself becomes a soft vessel for light. Few bioluminescent organisms have had a scientific afterlife as influential as Equorea victoria. What began as the study of a glowing marine animal became a turning point in modern science. Through its proteins, A jellyfish from the ocean helped researchers see life in new ways far beyond the sea. Equoria victoria lives off the west coast of North America. That geographic detail matters because this famous jellyfish is not just an abstract laboratory source. It is a real marine animal from a real coastal environment, and from those waters came one of the most important biological discoveries of the modern era. When Equoria victoria is disturbed, the edge of its body can glow green. This makes the animal seem simple at first, almost as though it is just a jellyfish that lights up. But that green ring is actually the visible result of a much deeper chain of chemistry and energy transfer happening inside its tissues. The history of jellyfish bioluminescence shows how a natural curiosity can become a basic scientific tool. Something first noticed as a glowing marine phenomenon eventually helped transform cell biology, genetics, and medical research. It is one of the clearest examples of pure observation in nature leading to major scientific progress. Fluorescence is different from bioluminescence because fluorescence needs incoming light, while bioluminescence creates light chemically. In simple terms, fluorescence changes light that already exists, but bioluminescence makes fresh light from a reaction inside the organism. That difference is very important when scientists study glowing systems. Some jellyfish are especially famous because they combine both of those phenomena in one optical system. They do not only produce light chemically, they also reshape that light through fluorescent molecules, which changes what the glow looks like to the eye. The scientific value of jellyfish light did not come mainly from brightness. It came from the stability and usefulness of the proteins involved. In science, a dim but reliable molecule can be more important than a bright but unstable one, and the jellyfish proteins turned out to be remarkably useful. GFP became important partly because it can produce a fluorescent signal in many other cells without needing a long list of extra components added from outside. That made it practical for researchers. Once the gene for the protein was inserted into cells, the cells themselves could make the marker. That simple usefulness helped a jellyfish protein spread across biology laboratories around the world. Scientists could attach it to genes or proteins they wanted to watch, and then track where those targets appeared inside living cells. A marine glow became a universal research language. In this way, one luminous system from the ocean moved into medicine, neuroscience, and genetics. The original glow belonged to a jellyfish, but the scientific usefulness of its proteins reached into many fields far removed from marine biology. Light from one animal helped reveal the hidden inner life of many others. Squid show several different uses of bioluminescence, including camouflage, signaling, and defense. This makes them especially interesting to study because they do not represent only one narrow function. Different squid lineages use light in different ways depending on where they live and how they survive. The vampire squid produces bright blue bioluminescence of the tips of its arms. In the deep sea, that localized glow can be startling and dramatic. The animal does not need a large, bright body display to make an impression. Even small points of light placed in the right spots can be powerful. The vampire squid can also release clouds of glowing fluid when threatened. This is a very different use of light from a fixed lure or a camouflage pattern. Instead of keeping the glow attached to its body, the animal throws light outward into the surrounding water. That glowing cloud functions like a luminous version of an ink cloud. Ordinary squid ink hides an animal by darkening the water. The vampire squid's version changes the idea completely by making the water light up instead, creating confusion in darkness rather than darkness in light. Unlike many active squid hunters, the vampire squid is a gentle scavenger. It does not match the dramatic image its name suggests. This makes its bioluminescent defenses even more interesting, because such strong-looking displays are not limited to fierce predators. That means a dramatic bioluminescent defense can belong to an animal that is not an aggressive hunter at all. A glowing display does not always tell us that a creature is dangerous in an offensive sense. Sometimes it simply tells us that evolution found an effective way for a vulnerable animal to survive. Some shrimp use light even more explosively by ejecting luminous secretions. Their defense is not a steady glow and not a delicate flash. It is a sudden release of glowing material into the water around them, turning the space itself into a defensive display. In some NOAA descriptions, a deep-sea shrimp can effectively vomit light near its mouth. The phrase sounds dramatic, but the function is practical. By releasing a bright, distracting burst where a predator is focused, the shrimp buys itself a moment to escape. A luminous secretion can distract a predator just long enough for escape. That small time window can be everything in the deep sea. A fraction of a second of confusion may separate survival from capture. Copepods are another group with many luminous species. These tiny crustaceans are easy to overlook because of their size, but they are major players in the ocean. Their light reminds us again that small organisms can have large ecological importance. Luminous copepods are widely distributed in the world's waters. This broad distribution shows that bioluminescence is not restricted to one isolated habitat or one rare lineage. It appears in many marine environments and across many forms of drifting life. The genera Purama and Matridia are notable luminous copepod groups. Naming them may seem technical, but it helps show that bioluminescence is studied in detail, down to particular branches of tiny animals. The world of living light is not vague. It has fine structure and many separate evolutionary stories. Ostracods are small crustaceans that also include bioluminescent species. These tiny animals have bodies enclosed in shell-like coverings, yet even within such small forms, evolution has produced ways to make light. Bioluminescence does not require a large or dramatic body plan. Because ostracods are so small, they show once again that powerful visual signals can come from tiny bodies. The effectiveness of light does not depend only on body size. In darkness, even a very small luminous source can matter if it is used in the right way. Among crustaceans, bioluminescence is especially prominent in copepods, shrimps, and ostracods. That concentration tells us that light has been useful across several branches of small marine arthropods. These are not isolated accidents, but repeated solutions appearing in related groups. Some worm-like or larval forms on land are bioluminescent, even when the adults look very different. This is important because it shows that light can be strongly tied to a life stage, not just to a species as a whole. The glowing body may belong to the juvenile form rather than the mature one. Australian glowworms are not actually worms, and they are not fireflies either. The name describes appearance rather than true biological identity. This is common in nature, where familiar words stay attached even after science reveals something more precise. Those Australian glowworms are the larvae of certain flies. That fact changes how we imagine them. Instead of thinking of them as mysterious worm-like lights, we can understand them as an insect life stage, using glow as part of a very specific feeding strategy. In caves and grottos, glowworm larvae use light to attract prey towards sticky threads. The light is not just decorative. It works as part of a hunting system, drawing small flying animals toward a trap hanging in darkness. In some well-known populations, the prey of cave glowworm larvae includes ghost midges. This tells us that the light has a real ecological target. The glowing display is part of an interaction between predator and prey, not just a general illumination of the cave. The hanging threads of glowworm larvae make cave ceilings function almost like suspended fishing nets lit by blue-green lamps. It is a powerful image, but also a useful biological one. The glow attracts, the threads capture, and the cave becomes a carefully arranged feeding environment. British glowworms belong to a beetle, not a fly. So even though two very different organisms share the common name glowworm, they come from separate branches of insect life. Their similar glow reflects similar usefulness, not close identity. In the British glowworm Lampyris noctiluca, the adult female climbs to a higher point and shines upward to attract flying males. Her light is part of courtship rather than feeding. This is a very different use of bioluminescence from the prey-luring larvae in caves. Adult female Lampyris noctiluca usually live only a few weeks, long enough mainly to mate and lay eggs. That brief adult life means the glow is closely tied to one urgent biological task. The light is not a long-term everyday feature. It is part of the short reproductive window. Because the adult female's life is so short, her glow is tightly linked to reproduction rather than general survival over time. In other words, the light is used at a very specific moment in the life cycle, when attracting a mate matters more than almost anything else. Some terrestrial bioluminescent species glow in eggs, larvae, and pupae, as well as in adulthood. This shows that light can appear across multiple stages of development. It is not always restricted to one phase, and its function may shift as the organism changes form. Railroad worms are not worms either. They are the larval forms of certain beetles. Like glowworms, they remind us that common names often describe appearance, while biology reveals a more complex and often more interesting identity. Railroad worms carry rows of green lights along the body. These repeated points of light give them a striking, segmented appearance. The pattern makes them look almost engineered, like a series of tiny lamps placed with precision. They also have red lights near the head, which is unusual in bioluminescence. Because red bioluminescent light is much less common than blue or green, railroad worms stand out as an example of nature exploring a rarer optical path. The diversity of glowing larvae shows that bioluminescence can be strongly tied to developmental stage. Larval life often involves different risks, behaviors, and habitats than adulthood. So it makes sense that light would sometimes be useful early in life, even if its role changes later. Deep-sea anglerfish are among the most recognizable bioluminescent fishes. Their silhouette, with a small glowing lure in front of the head, has become an icon of deep-sea adaptation. Yet behind that familiar image is a carefully evolved strategy shaped by darkness and scarcity. In many anglerfish species, only the female has the prominent glowing lure. This reflects a strong difference between the sexes, with the larger female taking on the visible predatory role. The light organ is not simply a species trait spread evenly across every individual. In some anglerfish species, females can reach about 20 centimeters, while males are much smaller. That size difference matters because it helps explain why the female carries the better-known hunting anatomy. The luminous lure belongs to the larger, more visibly predatory body plan. The anglerfish lure works because prey in darkness may investigate a small, moving light as if it were food. In a place where clues are scarce, even a faint signal can become irresistible. The prey's normal curiosity becomes the predator's advantage. Dragonfishes are another famous deep-sea group associated with bioluminescent hunting. Their use of light shows that predatory bioluminescence did not evolve only once. Different fish groups independently found ways to turn darkness into an advantage. Some dragonfishes use light to illuminate prey directly in the deep sea. This is a subtle but important distinction from luring. Instead of inviting prey closer, the fish can briefly reveal what is nearby, using light more like a search tool. The black dragonfish is one of the larger animals associated with burglar alarm-style luminous defense or display. This shows that even within one broad group, the meaning of emitted light can vary. A signal can shift from offense to defense depending on context and species. Stoplight lucijaws are especially unusual because they can make use of red bioluminescence. In the deep sea, where most available light is blue, that is an extraordinary specialization. It allows these fish to operate in a visual range that many neighbors do not use well. The stoplight lucijaw begins with blue light, but uses a protein to re-emit it as red. This means the final visible signal is transformed after the initial light is made. It is an elegant example of biology modifying its own optical output. Red bioluminescence is useful in the deep sea because many animals there do not detect long red wavelengths well. A predator able to use red light can therefore gain a kind of private view of its surroundings. It is like carrying a lamp that others nearby can barely notice. That makes red light a kind of secret illumination channel for certain predators. In evolutionary terms, this creates a sensory advantage. The animal can see more without revealing as much to the prey around it. Railroad worms evolved a similar red light trick on land, showing that unusual bioluminescent colors are not limited to the sea. This parallel is especially interesting because it suggests that very different organisms in very different settings can arrive at surprisingly similar optical solutions. Flashlight fish carry luminous organs beneath their eyes. This placement is important because it turns the face of the fish into a focused signaling area rather than making the entire body glow. In dark water, a small light positioned near the eyes can be useful for directing light outward, communicating with neighbors, or helping the fish control exactly how it appears. Those lights can help flashlight fish remain coordinated with nearby companions during nighttime schooling. In darkness, it is much harder for animals to keep track of one another using body shape alone. A small glowing organ can act almost like a moving marker, helping the fish maintain spacing and move together more smoothly as a group. Some deep-sea fish use light to help locate mates, not just prey. In a dark environment where individuals may be widely scattered, finding another member of the same species can be a major challenge. Light gives these animals a way to signal presence and improve the chances of meeting the right partner in the vastness of open water. In some species, the placement of light organs on the body can work like a recognizable pattern. Instead of only producing a general glow, the animal may display light in a specific arrangement that helps others identify it. In deep darkness, that pattern can serve almost like a visual signature. The cookie-cutter shark is a bioluminescent shark, not a typical bony fish. That makes it especially interesting because it shows that luminous evolution is not limited to one narrow kind of marine vertebrate. Even sharks, with their ancient lineage and very different body design, became part of the story of living light. Light organs cover much of the cookie-cutter shark's lower surface, except for the fins and a dark collar near the throat. This distribution matters because it creates a body-wide luminous effect rather than a single isolated light organ. The pattern suggests the glow plays a role in how the shark appears as a whole when viewed from below. The underside of the cookie-cutter shark glows bright green. In deep or dim waters, that color can blend with the faint light filtering down from above. The glow is not simply decorative. It changes how the shark is seen in the water column and may help it become less obvious until the last moment. The dark collar of the cookie-cutter shark may create visual contrast that helps lure prey close enough for attack. Instead of making the whole underside evenly bright, the shark presents one darker region that may stand out against the glowing body. That contrast may mislead larger animals and draw them into range. Cookie-cutter sharks are ectoparasites that cut round plugs of flesh from much larger animals. This feeding method is unusual and very precise. Rather than swallowing whole prey, they take small circular bites from animals many times their size, leaving behind the well-known cookie-shaped wounds that give them their common name. These sharks are generally caught at night and are thought to move upward from deeper water after dark. This upward movement suggests that depth and darkness both shape when and where their light-based strategies are useful. The shark is not simply glowing in one fixed zone of the ocean, but moving through the water column with daily rhythm. Some deep-water squalliform sharks are also bioluminescent. This broadens the picture beyond a single famous shark species. It shows that the ability to glow has appeared in more than one shark lineage, meaning the usefulness of light reached across different evolutionary branches of cartilaginous fishes. That means sharks, despite their ancient and often fearsome reputation, belong to the same wider luminous story as many other deep-sea animals. They are not separate from the world of glowing jellyfish, fishes, and plankton. They are participants in the same dark environment where light becomes a key survival tool. Many deep-sea sharks have vision tuned toward blue wavelengths. This makes sense in an ocean world where blue and blue-green bioluminescence are especially common. Their eyes are adapted not to every possible color, but to the wavelengths most likely to matter in their environment. The depths at which sharks live influence which wavelengths their photoreceptors are best suited to detect. In other words, their vision is shaped by habitat. The light available at different depths, including bioluminescent light, helps determine what kind of visual sensitivity is most useful. The optical environment of the deep sea is partly shaped by the light animals themselves create. That is a fascinating idea, because it means deep-sea creatures are not only adapting to darkness, they are also adapting to a world where other organisms actively produce the main visible signals. Some fish use luminous bacteria, while others make light using their own cells and chemistry. This tells us there is no single fish solution to bioluminescence. Different species reached the same broad ability through different biological routes, showing how flexible evolution can be. So the phrase bioluminescent fish does not describe one simple mechanism. It includes many different systems, each shaped by a species' ancestry, anatomy, and lifestyle. One fish may glow because of microbial partners, while another glows through its own specialized tissues. Deep-sea fishes may face pressure both to emit useful light and to hide from the light emitted by others. This creates a kind of visual arms race. A fish must use light effectively, but it must also avoid becoming too visible to predators or prey that can detect that same signal. For many people, fireflies are the most familiar example of land bioluminescence. Unlike deep-sea glowing animals, fireflies belong to everyday human memory in many places. Their lights are easy to notice, and because terrestrial bioluminescence is comparatively rare, they often become the symbol of the whole phenomenon on land. Firefly flashes are used largely in courtship communication. The glow is not random or purely defensive in most familiar cases. Instead, it is part of a mating system in which timing, pattern, and response all matter in helping individuals find suitable partners. Different firefly species use different flash patterns, rhythms, and flight behaviors. This means that not all glowing beetles are sending the same message. Each species can have its own timing rules, its own style of motion, and its own way of turning light into communication. Great Smoky Mountains National Park is especially famous for Photinus carolinus, the synchronous firefly. This species is well known because large numbers of individuals can coordinate their flashes in a striking shared rhythm. The display is so distinctive that it has become one of the most celebrated natural light events on land. In this species, many individuals in one area flash together and then pause together in darkness. The pattern is not just that they glow often, but that they do so in coordinated bursts. That shared timing creates an effect that feels far more organized than the scattered flashes of other firefly species. The synchronous display of Photinus carolinus is often described as a series of about 5 to 8 yellow flashes, followed by a dark pause lasting about 8 to 10 seconds. This rhythm gives the event its memorable structure. The repetition of clustered light and silence makes the forest seem to breathe in pulses. The large synchronous displays usually begin at full dark, around 9:30 a.m. at night, and can continue until about midnight. This timing means the spectacle belongs to a fairly narrow window of evening darkness. The light show is tied not only to season, but to the quiet hours when the environment is dark enough for the flashing to stand out fully. The mating season for the Smokies' synchronous fireflies lasts only about 2 to 3 weeks each year. That short season adds to the special nature of the event. The display is not available all summer long, but appears briefly, tied to the biological timing of the species. The peak timing of the display shifts from year to year, depending on conditions such as temperature and soil moisture. This means the fireflies are responding to the environment in a very sensitive way. Their timing is biological, but it is also shaped by the weather and the condition of the forest floor. Heavy rain can interrupt nightly flashing activity in synchronous fireflies. Even if the season is right, the display still depends on suitable conditions on a given night. Rain changes the behavior of the insects and can reduce the coordinated visual effect that observers hope to see. Cool temperatures below 50 degrees Fahrenheit can reduce the number of active fireflies in the Smokies. This shows that their luminous behavior is strongly connected to body function and environmental warmth. If the conditions are too cool, fewer insects participate, and the display weakens. Conbury National Park also hosts synchronous fireflies for about 2 weeks, between mid-May and mid-June. This tells us that the phenomenon is not limited to one famous place, though it remains rare. Different habitats can support similar luminous behavior when the right species and conditions come together. There are more than 2,000 firefly species worldwide, but only 3 synchronous species are found in North America. That contrast makes full synchronization especially unusual. Fireflies are diverse, but this dramatic kind of shared flashing belongs to only a small fraction of them. Complete synchronization is a rare ability among North American fireflies. Most species do not coordinate in that way. This rarity helps explain why synchronous species attract so much attention from scientists and the public alike. Firefly species do not all glow with the same color or the same rhythm. Some have different pulse spacing, others different brightness or timing, and some differ in flight behavior as well. Their light is not a single standard design, but a diverse collection of species-specific signals. Some firefly species use a double flash instead of a longer sequence. This shows that even small changes in timing can carry meaning. A brief pair of flashes can be as important as a long cluster if the receiving insect recognizes it as the correct signal. In Great Smoky Mountains National Park, Photinus macdermati is known as a double flash species. This gives a clear example of how neighboring luminous insects can still communicate in very different styles. Even in one broad region, fireflies may be speaking with different visual patterns. The genus Photuis includes predatory species that mimic the flash responses of females of other species. This is one of the most striking examples of deception in bioluminescent communication. The signal looks like an invitation, but it is actually a trap. Predatory Photuis females can lure in males from other species and then eat them. In this case, light is being used not for honest courtship, but for manipulation. The same visual language that usually brings mates together becomes a weapon of predation. So, firefly bioluminescence can support both romance and deception, depending on the species. In one lineage, the flash is a courtship signal. In another, that same kind of signal can be copied and turned against the one who trusts it. Some firefly viewing events are so popular that parks manage access through lotteries. This is a sign of how rare and impressive coordinated bioluminescent displays are on land. Human interest becomes so intense that access itself must be controlled. In 2019, nearly 29,000 people entered the lottery for synchronous firefly viewing at Elkmont. That number shows how strongly people are drawn to natural light displays, especially when they are seasonal, fragile, and difficult to predict. It also shows that a small insect can shape human behavior on a large scale. That level of public demand shows how unusual coordinated natural light displays are on land. We are surrounded by artificial lights, yet people still travel, wait, and plan carefully to witness flashes made by insects in the dark. There is something deeply compelling about seeing light emerge directly from living bodies. Unlike surface marine bioluminescence, firefly displays are often species-specific messages rather than generalized stress flashes. A glowing wave is often the result of disturbance in plankton-rich water, but a firefly flash is more often aimed at another firefly and shaped by communication rules. Land bioluminescence is rare enough that a forest full of synchronized fireflies becomes one of the clearest terrestrial parallels to glowing surf. In both cases, many small organisms create a larger scene of shared light. But on land, that kind of luminous gathering is far less common. British glowworms show that not all glowing beetles fly or flash in the same way as classic fireflies. The familiar image of a flashing insect in motion is only one version of luminous beetle life. Other species use light more quietly and from a fixed position. In some glowworm systems, the adult female is wingless and advertises from vegetation instead of flying. Her strategy is based on staying visible rather than moving through the air. The glow becomes a beacon rather than a traveling signal. The diversity of luminous beetles shows that bioluminescent courtship has evolved in more than one physical form. One species may signal while flying, another while perched, and another during a different life stage entirely. The shared feature is light, but the body plans and behaviors around it vary greatly. Because their flash patterns are species-specific, firefly signals are often described as a kind of visual language. This comparison is useful because it captures the role of timing, recognition, and response. The flashes carry information, and the meaning depends on who is watching. Terrestrial bioluminescence is much rarer than marine bioluminescence, which is one reason fireflies stand out so strongly in human culture. On land, glowing life is unusual enough to feel memorable and almost magical. In the ocean, by contrast, light production is far more common, especially in darkness. Fungi are one of the few major known animal groups known to include naturally bioluminescent species. This expands the story of living light beyond animals and microbes. It shows that even organisms rooted in decay and nutrient cycling can evolve the ability to glow. About 75 fungal species are known to give off light. That number is not enormous compared with the diversity of fungi as a whole, but it is enough to show that glowing mushrooms and luminous fungal growth are real recurring phenomena, not isolated accidents in nature. Most known bioluminescent fungi live in tropical rainforests. That setting makes sense because tropical forests are warm, humid, and full of decaying organic material, which is where many fungi thrive. In such dark and moisture-rich places, even a faint glow can become noticeable at night. The glow of many bioluminescent fungi is typically green. Green is not the dominant color of marine bioluminescence, So fungal light immediately adds a different visual note to the larger story of living light. In a dark forest, that green tone can make rotting wood or mushroom tissue appear softly lit from within. One leading idea is that fungal light attracts insects that may then help disperse spores. If this is correct, the glow is not only a chemical curiosity, it becomes part of the fungus's reproductive strategy, helping spread its offspring through the movements of visiting animals. That means fungal bioluminescence may function less like defense and more like advertisement. Instead of warning away attackers or confusing predators, the light may be inviting attention. In ecological terms, that is a very different use of light from many marine examples. Bioluminescent fungi show that glowing life is not limited to moving organisms. A fungus can stay rooted in place and still use light effectively. This reminds us that bioluminescence does not always need motion, pursuit, or active signaling to matter. Their light can make rotting wood appear to shine because the fungus lives within the decaying material. To someone walking through a dark forest, the wood itself may seem to glow. But the source of that light is actually fungal growth inside or across the surface of the decomposing matter. The historical term foxfire refers to the glow of certain fungi on wood. Long before modern science explained the chemistry, people noticed this strange light in dark forests. The name carries the memory of a time when luminous wood felt mysterious and almost supernatural. Fungal bioluminescence is one reason decaying forest matter can occasionally appear illuminated at night. That creates a very different emotional and ecological picture from ocean waves or firefly flashes. Here, the glow is tied to decomposition, moisture, and slow biological recycling. Unlike fireflies, fungi do not use moving flash codes, the courtship. Their light is not an active signal sent by a traveling animal. It is stationary, continuous, or gently persistent, and must serve its purpose without the help of flight, pursuit, or rapid timing patterns. Any function of fungal light must therefore work through a fixed luminous patch in the environment. This changes the whole logic of signaling. A glowing mushroom cannot chase, respond, or reposition itself, so whatever the light does must be accomplished by staying visible in one place. Because many bioluminescent fungi live in humid forests, their glow is often associated with damp, decomposing habitats. These are places where life is being broken down and returned to the ecosystem. The light, in that sense, belongs not only to beauty, but also to the quiet work of decay and renewal. The existence of glowing fungi also shows that bioluminescence evolved outside the animal kingdom. This is an important point because it expands the phenomenon beyond animal behavior alone. Living light is not confined to creatures with eyes, muscles, or nervous systems. Fungal light is much less familiar to most people than firefly flashes, partly because it is dim and often found in specific habitats. Many people may live their whole lives without seeing glowing fungi in person. Their rarity in everyday experience makes them feel especially hidden. Bioluminescent fungi are often discovered by careful night observation rather than by obvious daylight signs. In daylight, the fungus may appear ordinary or be hidden among dark forest textures. Only in darkness does its special quality reveal itself clearly. The green tone of many fungal species contrasts strongly with the blue dominance of marine systems. This difference reminds us that the color of bioluminescence is shaped by environment and function. A forest fungus does not face the same optical conditions as an animal drifting through seawater. Fungal light makes clear that not all luminous systems are built for long-distance transmission through water. In the ocean, color choice is strongly shaped by what wavelengths travel best. In a forest, the problem is different, and so the glow can take on different colors and uses. Because fungi are decomposers, their glow connects bioluminescence to nutrient recycling, as well as to predation or courtship. This gives fungal light a different ecological meaning from the light of hunting fish or signaling beetles. It belongs to a world of breakdown, absorption, and ecological return. A glowing mushroom and a glowing fish therefore represent very different solutions to very different problems. One belongs to a stationary decomposer in a forest, and the other to an active animal in water. The shared feature is light, but the evolutionary logic behind that light can be completely different. Bioluminescence has evolved many times independently rather than arising from one single glowing ancestor. This repeated origin tells us that making light has been useful often enough for evolution to invent it again and again. Different lineages found separate paths to the same broad result. Research has suggested that bioluminescence in corals originated at least 540 million years ago. That date pushes the history of living light very deep into the ancient past. It means bioluminescence is not a recent novelty, but part of a very old evolutionary story. That finding also pushed the origin of coral bioluminescence much earlier than many people once thought. Scientific understanding often changes when new methods or broader comparisons reveal deeper roots than expected. Bioluminescence keeps surprising researchers in that way. The widespread distribution of bioluminescence across bacteria, protists, fungi, and animals is strong evidence of repeated evolutionary invention. These groups are far too different for one simple glowing origin to explain everything. Instead, nature seems to have rediscovered light many times. Similar functions can evolve in unrelated luminous organisms, even when the underlying chemistry differs. This is a classic pattern in evolution. Different bodies, molecules, and lineages can still arrive at similar survival strategies when they face comparable environmental challenges. Blue light dominates marine evolution partly because seawater transmits shorter visible wavelengths more effectively. In practical terms, blue and blue-green travel farther than red in water. So organisms that rely on light in the ocean often benefit from producing colors that move efficiently through their environment. Red bioluminescence is rare, partly because red light disappears quickly in water and is hard to exploit unless both production and detection evolve together. A red signal is only useful if the organism can make it, and also benefit from a world where others do not see it well. That makes it a more specialized evolutionary path. Counter-illumination likely evolved repeatedly in unrelated marine animals living in open water. The problem it solves is common: how to hide your silhouette when there is faint light above and open space all around. Because that problem appears in many lineages, the solution appears again and again. The deep sea favors light-based traits because darkness makes even small optical differences biologically important. In brighter environments, many signals compete with sunlight and background detail. In darkness, however, even a weak flash can carry enormous importance. Scientists long underestimated how common deep-sea bioluminescence was, because early estimates were based mostly on qualitative impressions from submersibles. Those observations were valuable, but they did not always provide precise measurement. As a result, the true scale of glowing life in the deep sea took time to understand. A quantitative MBARI study published in 2017 helped replace those earlier impressions with measured estimates. This matters because science becomes stronger when broad ideas are backed by systematic counts and careful analysis. The result was a clearer picture of just how widespread living light really is. Modern understanding of bioluminescence depends heavily on remotely operated vehicles because many luminous animals are damaged or missed by nets. Fragile, gelatinous bodies, delicate tissues, and behavior-based light displays are often best observed intact in the water. technology allows scientists to study them more gently. Recording a bioluminescent response can be tricky because bright vehicle lights may alter animal behavior. If the observer floods the scene with artificial light, the very phenomenon being studied may change or disappear. This makes the science technically challenging. Researchers therefore design low-light or specialized imaging approaches to study natural displays. They try to see the light without overwhelming it. In effect, the tools must become subtle enough to watch the deep sea on something closer to its own terms. The scientific study of bioluminescence sits at the meeting point of chemistry, cell biology, ecology, behavior, and optics. That is one reason the field is so rich. To understand a glow fully, scientists often need to know not only what molecules are involved, but what the organism is doing, who it is interacting with, and how the light travels. Some luminous systems were first investigated simply because their glow was visually striking, yet later turned out to have medical or biochemical value. This is one of the quiet lessons of natural history. Something that first attracts attention for beauty may later transform science. The story of GFP is a clear example of basic natural observation leading to a major research tool. People did not go to a jellyfish expecting to change cell biology, but careful attention to one natural light system opened the door to entirely new ways of seeing inside living cells. Bioluminescence research also helps scientists understand how animals communicate in places where sound, color, or body shape are hard to use. In the deep sea especially, light may become the most practical visual language available. The study of glowing creatures is therefore also the study of communication under extreme conditions. In deep ocean ecology, light can play a role somewhat comparable to birdsong in forests or scent trails on land. It becomes one of the main channels through which organisms announce themselves, attract mates, warn enemies, or deceive prey. The medium changes, but the importance of signaling remains. Each new luminous species studied can reveal a different evolutionary route to making light useful. No single species explains the whole phenomenon. Every new case adds another variation in chemistry, anatomy, behavior, or ecological purpose. Equirin from jellyfish became an important calcium indicator in research. This means a molecule that originally helped produce light in an animal could later help scientists study calcium activity inside cells. A natural glow became a window into cellular processes. GFP became a standard marker for tracking gene expression in living cells. Researchers could link it to genes and then watch where and when those genes became active. In this way, glow became evidence of life happening at the molecular level. Scientists can attach GFP to proteins to see where those proteins move inside cells. That simple idea has enormous power. Instead of guessing where a protein goes, researchers can observe its path and location more directly in living material. GFP made many previously invisible biological processes visible in real time. This changed not only what scientists knew, but how they could know it. A process that once had to be inferred could now sometimes be watched as it unfolded. Researchers have specifically used GFP to observe nerve cell development. That means a protein borrowed from a luminous marine organism helped scientists study one of the most complex systems in biology, the developing nervous system. The journey from jellyfish to neuroscience is remarkable. GFP has also been used to study how cancer cells spread. This is one more example of a natural light system becoming part of serious medical investigation. A glow first evolved in one context can become useful in understanding disease in another. Part of GFP's appeal is that the protein itself can generate a useful signal when expressed in other organisms. It does not require an overly complicated supporting system to be valuable in the lab. That practicality helped it become one of the most widely adopted tools in biology. Bioluminescent and fluorescent proteins are now central tools in molecular biology laboratories around the world. They are no longer rare specialties. They have become part of the standard scientific toolkit for making invisible processes visible. Studying natural luminous proteins has also inspired new engineered imaging tools. Scientists do not only copy nature directly. They learn from it, modify it, and build new technologies based on its principles. Natural light systems become starting points for innovation. The path from glowing jellyfish to advanced microscopy is one of the clearest examples of a natural phenomenon shaping biotechnology. It shows that close attention to the living world can lead not only to understanding, but also to new scientific capabilities. Because many deep-sea animals use blue light, designers of scientific instruments often tune sensors to wavelengths common in marine bioluminescence. The biology influences the tools. To study the light properly, the instruments must be built around the light most organisms actually produce. Bioluminescence research also helps engineers think about low-light signaling and detection systems. Nature has been solving optical problems in darkness for millions of years. By studying those solutions, people can gain ideas for human technology. Scientists interested in host microbe partnerships use luminous squid systems to understand how beneficial bacteria colonize tissues. The bobtail squid is not only a story about camouflage. It is also a model for how an animal selects, accepts, and manages helpful microbes. The bobtail squid symbiosis has become a model for how animals distinguish useful microbes from the many others in the environment. That makes the system valuable far beyond bioluminescence itself. It helps scientists think about recognition, immunity, and cooperation in living bodies. Flashlight fish and anglerfish show that biological lighting can be anatomically directed, filtered, and reflected, much like light from a carefully designed lamp. The glow is not always allowed to spill out in every direction. In many species, body structures help shape where the light goes, how focused it becomes, and how useful it is to the animal. The study of private red bioluminescence in deep-sea fishes helps scientists understand how visual secrecy can evolve in a sensory arms race. If one animal can use a color of light that many nearby animals barely detect, it gains a hidden advantage. That means the deep sea is not only a place of darkness, but also a place of quiet visual strategies that are partly concealed from most eyes. Optical adaptations in luminous sharks help connect sensory biology with habitat depth. A shark living in deep water does not need the same visual system as one living near the surface. Its eyes, its light environment, and its behavior all become linked, showing how vision and bioluminescence shape each other over time. Fungal and insect bioluminescence are especially useful for comparison because they show very different ecological uses of light. One may involve a stationary glow in a damp forest, while the other may involve moving flashes in courtship or predation. Comparing them helps scientists see that the same broad phenomenon can serve very different purposes. In practical research, glowing organisms are often easier to track in darkness than non-glowing ones. A luminous body may reveal movement, timing, or behavior that would otherwise be hard to follow. That can simplify some observational studies and help scientists notice patterns they might have missed in total darkness. Bioluminescence has become both a subject of discovery and a tool for making other discoveries. Scientists study glowing organisms for their own sake, but they also use luminous molecules to study cells, tissues, and disease. In that way, living light helps us understand both itself and many other parts of biology. Human encounters with bioluminescence often depend more on darkness than on rarity, because even a modest natural glow can disappear in artificial light. A shoreline, forest, or cave may hold luminous life that goes unnoticed if the night is too bright. Sometimes the phenomenon is present, but the world around it is simply too illuminated for us to see it. Shoreline bioluminescence is often easiest to notice where wave action constantly disturbs plankton-rich water. The movement of the surf repeatedly triggers tiny flashes, making the water seem alive with light. The shoreline becomes a place where ordinary motion reveals an extraordinary hidden biology. Night dives and submersible trips show that many bioluminescent animals are invisible until they react. A creature may remain unseen in the dark until movement, contact, or disturbance causes it to flare into view. This means much of the luminous life in the ocean is not obvious at first glance, but appears only in moments of response. A glowing animal can be easier to notice than to identify, which is why deep-sea surveys often need video, specimens, and context together. Light can announce that something is there long before it tells us exactly what that something is. Recognition and understanding usually require slower, careful study. Some of the most dramatic marine light shows come from entire communities rather than from one especially conspicuous species. A glowing sea surface, for example, may be the combined work of countless microscopic organisms. In those cases, the beauty of the event comes from the scale of participation rather than from a single luminous body. On land, by contrast, people often focus on individual insects or local gatherings of a single species. A firefly flash is often noticed one body at a time or in a particular patch of forest. The luminous experience on land is usually more local and personal, while in the sea, it can become wide and landscape-like. Tourism built around synchronous fireflies shows that luminous behavior can influence how protective areas are managed. When large numbers of visitors gather to watch a delicate biological event, parks have to think carefully about timing, access, and preservation. The glow of insects can shape human planning in very real ways. The popularity of firefly viewing also means habitat disturbance can become a concern for glowing species. Trampling, excess light, and crowd pressure can all affect sensitive environments. A natural wonder can attract admiration, but it can also create pressure that the habitat must somehow absorb. Because firefly timing depends on weather and soil conditions, climate change and land use change can alter when displays occur or how strong they become. Even subtle shifts in temperature or moisture can affect emergence and activity. This makes bioluminescent events not only beautiful, but also sensitive to environmental change. Marine blooms that create spectacular light are also part of complex coastal systems involving nutrients, water stability, and plankton dynamics. A glowing event at the shore may reflect a whole network of conditions building over time. What appears as a simple blue shimmer may actually be the visible surface of a much larger ecological process. Not all bioluminescent displays are equally predictable, which is why some famous viewing events are managed around estimated peak dates rather than fixed calendar dates. The organisms follow biology and weather, not human scheduling. This uncertainty is part of what keeps bioluminescent events both scientifically interesting and emotionally compelling. A luminous display can be both scientifically informative and aesthetically striking at the same time. Researchers may see ecological clues, chemical systems, or behavioral patterns in the glow, while other observers simply experience wonder. Bioluminescence lives comfortably in both worlds. Bioluminescence is one of the few natural phenomena that directly links invisible chemistry to visible behavior in real time. A molecular reaction becomes something the eye can actually witness. That direct path from chemistry to visible action is one reason bioluminescence feels so immediate and memorable. Its repeated evolution across unrelated groups shows that making light has been a useful solution in many ecological settings. Whenever the environment rewards signaling, camouflage, deception, or attraction through light, evolution seems capable of inventing it again. The glow is repeated not because life copies itself exactly, but because the solution works. The ocean's darkness did not suppress visual evolution. Instead, it encouraged new forms of vision and signaling built around self-made light. In the absence of sunlight, animals found ways to become their own sources of visibility and meaning. On land, where darkness is more intermittent and visual backgrounds are often more complex, luminous signals are much rarer and often tightly specialized. Forests, grasslands, and open air create different visual problems than the deep sea. That is why terrestrial bioluminescence tends to appear in narrower and more specific roles. Bioluminescence can be defensive, deceptive, cooperative, reproductive, or useful in hiding, depending on the organism. A single glow may mean warning in one species, camouflage in another, and invitation in a third. The light itself is only part of the story. The purpose depends on the life around it. A glowing line of surf, a flashing firefly, a luminous shark belly, and a green mushroom all belong to the same broad biological phenomenon, but not to the same evolutionary history. They are united by light, yet separated by chemistry, ancestry, habitat, and purpose. That makes bioluminescence both one subject and many subjects at once. The study of bioluminescence keeps expanding because each newly studied glowing organism may reveal a new chemistry, a new behavior, or a new partnership. Scientists do not yet have one complete map of living light. Each discovery adds another piece to a much larger picture. Bioluminescence is not one single natural trick, but a vast collection of independently evolved living light systems spread across oceans, forests, caves, and the history of science. The more closely it is studied, the more it appears not as one story, but as a whole library of stories written in light. Equirin, the light-producing protein from jellyfish, became an important calcium indicator in research. That means a molecule first known for helping create a marine glow later became useful for studying how cells handle calcium signals. A light-making system from one organism ended up illuminating the inner activity of many others. The 2008 Nobel Prize in Chemistry recognized the discovery and development of GFP. That honor reflects how deeply one glowing protein changed modern biology. A natural molecule from a marine animal became important enough to reshape scientific practice across the world. The original glow of a jellyfish changed how scientists studied genes, cells, and disease. What once looked like a beautiful marine curiosity became a way to watch biology in action. That shift from observation to application is one of the quiet triumphs of scientific history. Some deep-sea jellyfish use luminous displays as defense, producing sudden patterns that can distract attackers. The light may interrupt the predator's focus just long enough to create an opening for escape. Even delicate bodies can rely on surprisingly powerful visual tactics. Scientists often use remotely operated vehicles to study gelatinous, luminous animals because net collection can damage their fragile bodies. If the animal is torn, compressed, or altered, its natural light display may be lost or distorted. Gentle observation is often the best way to understand how the glow truly works. Some jellyfish glows become visible only when the animal is disturbed. This means the animal may remain nearly unseen until something touches it, threatens it, or changes its surroundings. The light is not always a constant lamp, but sometimes a triggered response. The light in gelatinous animals often spreads through transparent tissue, making the whole body seem illuminated from within. This creates a soft and unusual visual effect, different from the point-like glow of a lure or a photophore. The body itself becomes a vessel for light. Some of the most iconic glowing animals are really tiny ecosystems in miniature, because their light comes from symbiotic bacteria living inside special organs. The animal and the microbes function together so closely that the visible glow is the product of partnership. In such cases, the light belongs to a relationship as much as to an individual. Some bioluminescent systems use brief flashes, while others create steady glows. This difference matters because timing changes meaning. A pulse can surprise, signal, or warn, while a steady light may attract, camouflage, or quietly mark a presence. Flash patterns can carry information in a way that resembles timing patterns in sound-based communication. The exact number of flashes, the spacing between them, and the pauses that follow can all matter. In that sense, bioluminescence can function almost like a visual rhythm. Much about the functions of bioluminescence is still unresolved because direct observation in the deep sea remains difficult. Scientists can watch, measure, and compare, but some behaviors are still hard to witness under natural conditions. There is still mystery in the glow. Many scientists think dinoflagellate flashes may help scare away small grazers. A sudden spark in the water could startle a tiny predator or make feeding less smooth and less safe. Even a microscopic organism can use a moment of surprise as protection. Another idea is that dinoflagellate flashes attract larger predators that attack the dinoflagellate's attackers. In that case, the light functions like a call for danger rather than a direct defense. The tiny organism uses the food web itself as part of its strategy. The dinoflagellate Pyrocystis fusiformis uses an internal biological clock that helps determine when it becomes luminous. This means the ability to glow is connected not only to chemistry, but also to timekeeping inside the cell. Even the microscopic light display can be shaped by daily rhythm. In Pyrocystis fusiformis, the chemicals associated with light production are made at dusk. That timing helps explain why some bioluminescent plankton are especially ready to flash at night. The organism prepares for darkness before darkness fully arrives. The glow of dinoflagellates is often easiest to see where the water surface is physically agitated. Motion becomes the key that unlocks the hidden light. A calm surface may appear ordinary, but a wave or wake can suddenly reveal a world of luminous response. Many dinoflagellate flashes are responses to movement in the surrounding water. The light is not random decoration. It is a reaction to physical change, triggered when the organism is disturbed by passing motion. Dinoflagellate bioluminescence is one reason nighttime surf can appear electric blue. The color seems almost unreal, but it comes from vast numbers of small cells, each answering the motion of the water in their own tiny way. The shoreline becomes a place where chemistry and movement meet. Bioluminescent fluid can act like a glowing smokescreen in water. Instead of hiding by making darkness, an organism can hide by creating confusion through light. This is a beautiful reversal of what we often expect from concealment. Because many deep-sea animals make light, there is strong selective pressure for camouflage against that light. In a glowing world, it is not enough to avoid sunlight. An animal must also avoid being revealed by the light of its neighbors and enemies. In the deep ocean, surviving encounters may depend as much on not reflecting light as on producing it. This is why ultra-dark skin, careful body design, and subtle positioning matter so much. Sometimes the quietest strategy is to disappear from the light of others. Bioluminescence among fungi, insects, plankton, fishes, and jelly-like animals shows that living light can be woven into almost every scale of life, from microscopic cells to larger predators. It can belong to drifting communities, solitary hunters, rooted decomposers, and brief courtship encounters. The glow changes shape with the life that carries it. And so bioluminescence remains a gentle reminder that the natural world is full of quiet inventions. In dark water, in damp wood, in still caves, and in soft summer air. Living things have learned to make light, for reasons as practical as survival and as intricate as communication. The more we learn, the more the darkness seems not empty, but softly inhabited by small and patient signals, continuing on in their own calm way.